Now we're going to do a crash course of the major organelles. This is going to go very quickly. If you need more information, you're welcome to go to your textbook or use the mastering system. The nucleus is probably the most complicated of the eukaryotic organelles. It is this major defining organelle, so let's break that down a little bit. This is a double layered membrane structure, so there's very few organelles that have two layers, and this is one that we're going to point out. So you have two layers of membrane that are just like the outer membrane, but obviously internal. In between those two layers of membrane, you have the nuclear lamina. The nuclear lamina is this major component down here. It's basically cytoskeleton fabric that provides strength to the nuclear membrane, allowing it to hold its shape and to protect the nucleus inside, the DNA inside. Inside the nucleus, you have dense genetic material, that is the chromosomes condensed into chromatin. It will then be spread out throughout the cell. Euchromatin will be accessible by that particular cell. Heterochromatin will not be accessible by that particular cell. Genes that the cell does not need to express, say for example a brain gene in the heart, would be heterochromatin, whereas muscle proteins for cardiac muscle would be active in the euchromatin. Within that, there's going to be an even denser region of overlapped chromatin, and that is what's called the nucleolus. That's all of the components necessary to produce ribosomes, all overlapping and in the same place. So that's the nucleus. Now things are going to need to get in and out of the nucleus, and it's protected by those two layers of membrane and the dense fabric. So you do have nuclear pores that will allow things to come in and out, like ribosomes and RNA, but otherwise the nucleus is a safe, protected space. Ribosomes are the protein factories. They're made by the nucleolus. They come in two types, bound and free. They're all the same thing, but are they attached to a membrane or are they free floating in the cytoplasm? So that could be attached to any membrane anywhere in the cell, and its job is to produce proteins. The most well-known bound ribosomes are going to be the ones that are attached to the rough endoplasmic reticulum, but bound ribosomes will make proteins that are destined to leave the cytoplasm. Okay, so they're going to go inside an organelle or outside of the cell. Why? Because they're attached to membranes, which are the transport systems that move everything around. Free ribosomes are going to make things that stay in the cytoplasm because they're not attached to any membrane, so they're not going to be transported. Now, prokaryotic ribosomes are smaller than eukaryotic ribosomes. You can see that here with the 70S versus the 80S. So we can target that for like disease-causing structures, but we do need to be careful because we do depend upon a lot of prokaryotes for our microbiome. The endoplasmic reticulum, these guys are biosynthetic factories. There's many different types of ER. You're expected to know the most common two, the smooth ER and the rough ER. You can see here the smooth ER is much more sort of branched, sort of loose structure. It's kind of looks like a fatty because it makes lipids. Okay, the smooth ER is a factory for lipids. The rough ER, since it has all of those protein-producing ribosomes attached, its job is to produce proteins. The smooth ER and the rough ER together make lipids and proteins, membrane. So this is your membrane factory. It's attached to the nuclear envelope, so we can get directions and materials back and forth, and then it will bubble off pieces as it makes membranes as a transport vesicle. The Golgi apparatus is your shipper processor, so things are going to go into the Golgi, and it's going to package it, modify it, and then send it wherever it needs to go. So things that leave the endoplasmic reticulum will go to the Golgi to be transported out of the cytoplasm or out of the cell. So if the factory is the ER, then Golgi apparatus is the mailroom sending it out. And these are what they look like in an actual cell. The more regular smooth ER because of the high lipid content, and the more regular linear rough ER for the organized protein control, and then the Golgi apparatus is this sort of folded pancake structure. As it goes through each of these layers, it gets progressively more modified with these vesicles then bubbling off. There are two special organelles that are made by the ER and the Golgi apparatus, the lysosome and the peroxisome. Lysosomes are digestive compartments. They're full of enzymes that are going to allow for things like digestion. They're going to allow for autophagy, that kind of thing. So we can see an example of the lysosome here. It's sort of your cellular stomach. The lysosome is produced by the Golgi body to break down old or malfunctioning components to be recreated into new. The peroxisome is produced by the smooth ER. The smooth ER has an additional function, including synthesis, and that is detoxification. And the peroxisome also aids in detoxification using hydrogen peroxide, which is why we call it a peroxisome. We used to think that these only occurred in plants, but we know that that's not true now. And the peroxisome also plays a role in catabolic breakdown of fatty acids. So the peroxisomes also help us digest fat, which is helpful. Vacuoles and vesicles, these are transport and storage components. Really the only difference is size. Vacuoles are large, vesicles are small. They're bubbles of membrane that are produced by a variety of structures, like the ER, and they're going to move around. Okay, so food vacuoles, contractile vacuoles, the large central vacuole of plants, they tend to be for storage, okay, pumping, that kind of thing. Whereas transport vesicles are going to move materials produced by the cell all over the place, really. The mitochondria, the powerhouse of the cell, might as well get it out of yourself now. Okay, this is another double-layered organelle. This is only our second double-layered organelle. So we have two layers of structure, which you can see in the picture on the left. The mitochondria is where cellular respiration occurs. So this is our major energy producer. It is a very special organelle, and we'll get to why later on. But the mitochondria is incredibly important because it is the main ATP factory. So all eukaryotic cells, for the most part, are going to be dependent upon mitochondria. There are a couple of exceptions. The chloroplast is the site of photosynthesis. The chloroplast is, again, a double-layered organelle, just like the mitochondria. And it is also special. We'll get into why later on. But it's a dynamic structure that moves around just like the mitochondria. It has its own DNA, just like the mitochondria, and it has tons of surface area, allowing for efficient chemical reactions. In the mitochondria, the surface area comes from the folding of the inner membrane, and in the chloroplast, the surface area comes from the folding of the thylakoid membranes, which we can see on the right. You need these surface areas because that's where the proteins are for production of ATP and energy capture, that kind of thing. So the mitochondria and chloroplasts are going to both be dependent upon that. Once you move beyond that into the actual cytoskeleton, we see that there are three different types of cytoskeleton, but overall, the cytoskeleton provides support and motility and regulation. It's a network of fibers that extend through the cytoplasm, and it is especially important in animal cells because we don't have cell walls. Okay, it provides anchorage for many organelles. It helps for assembly of structures. The largest of them, and the one that is rearranged and broken down and built up the most often, is the microtubule. The microtubule, which is composed of tubulin, is the key organelle builder for things like cilia and flagella, so lots of jobs here. But the microtubule is a hollow rod about 25 nanometers in diameter. It's the largest of these things. You have these specialized arrangements for many different structures, but we can see that the microtubule is absolutely essential for cellular motility. Motor proteins are going to work with the cytoskeleton, with the microtubules, so I should probably mention them here. Motor proteins are proteins that allow things to be moved around, because like vesicles, for example, they don't move by themselves. These proteins will move them around. How do they walk around? Well, they use ATP to bind to those microtubules. In animal cells, the microtubules are made by the centrosome. So the centrosome is what's known as our MTOC, or our microtubule organizing center. So the microtubules are absolutely essential, building these networks and anchoring these organelles and allowing for motility. Fungi and plant cells, they lack centrosomes. They don't need them. They have evolved ways to organize these without them, and we'll talk more about that when we get to how cells divide. The smaller components of the cytoskeleton, the microfilaments and the intermediate filaments, I'll give you one guess which one is in the middle. Okay, microfilaments typically are made of actin, and you might have heard that word before thinking about muscles. Our muscles are made of actin and myosin, so microfilaments are really important for us as animals. Microfilaments, they form structural networks. They're highly branched, able to make lots of connections. Important for cell motility, working with myosin. Pseudopodia, so like amoeboid movement, that kind of thing. In plant cells, they use myosin to flow material through their cytoplasm, so lots of involvement there. Anchorage of cells, muscle contraction, cell division, all important microfilament controls. The intermediate filaments are more permanent in structure. These things are like a twisted metal wire. They're not actually metal, they're keratin, like your fingernails, but the intermediate filaments are a very diverse class. They are more permanent fixtures of the cytoskeleton. Even after your cells die, the intermediate filaments often persist, and they reinforce cellular shape and cellular organelle location. The nucleus sits within a cage made of intermediate filaments fixed in location. The nuclear lamina is made of intermediate filaments. You get the idea here. Very important for maintaining cellular shape. The cell wall is something that animal cells don't have, but plants, animals, and bacteria do. They're going to be made of a variety of sugars. They might have some proteins associated with them, and there might be multiple layers depending on what type of thing that you're looking at. Most plants, for example, have at least 2 layers. People think of the cell wall like a brick wall, like you're not getting through, but it's a network of sugars. So like you can see on the right, for example, fungus is able to grow around and within the cell wall, and that's how symbiotic fungus, like mycorrhizae, spread out the roots of plants, allowing it to absorb more and more water. So the cell wall is structural, it's supportive, it prevents excessive uptake of water, it protects plants against the force of gravity, but it's not a brick wall. It's a permeable layer of complex polysaccharides and proteins that allow for structural support. The plasma membrane itself we'll get into more detail later on, but you should know that it's made of a double layer of phospholipids with the hydrophobic areas inside and the hydrophilic layers outside. We'll get into why. Well, we kind of talked about it before when we talked about a phospholipid, but you get the idea. Polar faces out, nonpolar faces in, creating a separate environment. Proteins allow for the flow of information and material across that plasma membrane, and outside of that plasma membrane, you're going to have a network of sugars and proteins called proteoglycans, so protein-sugar combo. Okay, the proteoglycans are going to work together with the proteins like fibronectin and integrin, which you can see here, to allow for communication between cells, cell-to-cell recognition, influencing cellular activity. The extracellular matrix is something that exists in all eukaryotic cells, but is much more elaborate in an animal cell because of the high need for cellular communication between our cells. Because we are motile, we need more communication. Plant cells have some ECM, but it's much more limited within what it does because most plants don't move. You get the general idea. And then between cells, our last major structure, we have our cellular junctions. There are 2 major categories of junctions. You have open junctions and closed junctions. Okay, open junctions, open flow, open connection. Okay, one of them you've seen already when we talked about plant cell walls, and that was the plasmodesmata. That is an open junction between cell walls that allow the free flow of small amounts of material. In animal cells, we don't have a cell wall, but we still need those openings. And so in an animal cell, it's called a gap junction, but it's the same idea. The gap junction is a small space that allows for the passive diffusion of ions and sugars and amino acids. Embryos, for example, are absolutely dependent upon gap junctions to facilitate chemical communication for development. The closed junctions are going to be those strong connections that are going to hold the cells together. And there's 2 types of closed junction. You have the tight junction and the desmosome. The desmosome is also called the anchoring junction. It fastens cells together into strong sheets like screws or rivets, and they're typically made of intermediate filaments of keratin to reinforce that structure, really strong connections. The tight junctions are formed sort of a continuous belts where the membranes have fused together. This prevents leakage and makes it watertight. So your stomach, for example, all of those cells of your stomach tissues are sealed together with tight junctions so that you don't leak stomach acid into your abdominal cavity and you don't digest your intestines. I think that's a good idea. I think we can agree. So that was our crash course of organelles. Feel free to go back and If you have any questions, please ask.